Sorry, I hit a button and escaped out of the presentation. Let me make sure it's recording. Are you recording? Yeah, okay. Back. Alright, so when I stopped, I was around here on this slide. So I want to just remind you that after fertilization, so again, the steps on the first part of the slide have occurred in the ovary. The dorsal protein, so not the axis of dorsal, but a protein called dorsal, is activated in the oocyte nuclei nearby the signal from the ventral follicle cells. Okay, so I want to stop here and just say that flies, especially early mutations that were discovered, these mutations in genes were named by the mutant phenotype. So, for example, normal flies have red eyes. But one day, there was a guy looking at a bunch of flies, and he found one that had white eyes. And so he named that mutation white, because the flies had white eyes. But, confusingly, the white gene is responsible for making red eyes. So it makes things a little complicated. To make things even more complicated, this person who discovered this gene named it dorsal. And that's because if you do not have dorsal, the gene, then everything in these follicle cells on the outside of the embryo all have dorsal phenotypes. They look like the whole thing is dorsal. There's no ventral. But that means that the dorsal protein is required to make things look ventral, right? It's a little complicated. Maybe you can rewind this part of the video and listen to it a couple of times. But the dorsal protein is required to make ventral phenotypes. And I know it's confusing, and I apologize for whoever discovered this mutation. It makes perfect sense when you're doing experiments in lab. When you suddenly see a white eye or an embryo where everything looks dorsal, you're like, oh, I'm going to name that gene white and that gene dorsal. But when you're trying to explain something to a class of students, I understand it's very confusing. So let's talk about the dorsal protein. The dorsal protein is a morphogen. It acts to induce different cell fates along the ventral to dorsal axis. Dorsal protein shown here is highest at the most ventral parts of the embryo and then gets lower and lower as you move more dorsally. So when you have, again, remember that idea, that concept of a morphogen I talked about on one of the first slides of this lecture. Those cells that receive the highest amount of dorsal down here are fated to become mesoderm. So that's cell fate number 1. If you cross a threshold, right here would be that threshold. Now you're making neuroectoderm, these yellow cells. And if you cross another threshold of dorsal protein concentration, now you're forming the dorsal part of the embryo. So dorsal is a true morphogen. It induces cell fates in a concentration-dependent manner along the ventral to dorsal axis. I want to show you another way to look at this. So here is that same picture from the last slide. High dorsal protein in the ventral part of the embryo. Again, sorry. These are the red mesodermal cells. The neurogenic, this is neuroectoderm. You have lateral ectoderm, dorsal ectoderm, and this structure called the amnioserosa, which is just a covering that forms around the embryo later. You can look at this. This is looking at what's called a transverse section. So basically, if you look at this left image and follow my mouse, I'm cutting the embryo right here and looking at what structures are formed. If you looked at it from the lateral view, this is the ventral, this is dorsal, you see that this is how the structure looks. You have the mesoderm and all these structures here. Remember, these are pole cells, right? They've been set aside early in development. So the dorsal gradient is what establishes all of these cell types in a concentration-dependent manner. So dorsal is really important for ventral patterning formation. So how does it do that? What are the effects of turning on dorsal in these cells? Well, those 16 cells right here at the bottom that receive the highest levels of dorsal become the mesoderm. All of the mesoderm comes from those cells to get that high level of dorsal protein. Dorsal can activate 2 key genes that I expect you to know, twist and snail. Twist is shown here. So dorsal turned on twist in these cells. And dorsal can also, the dorsal protein can also turn off genes that are required for cells to take a dorsal fate. It's getting more confusing now. Two dorsals on one sentence. So the dorsal protein can repress genes that are required to make cells take a dorsal fate. So it's promoting genes that promote ventral fate, and it's repressing genes that promote dorsal fate. These mesoderm cells, these twist-positive cells, are the first to undergo gastrulation. And so this is our first kind of dive into gastrulation in the fly. This process of this flat epithelium of cells forming this little lump inside and eventually pinching off to form a tube is called invagination. The invagination of the mesoderm creates a structure called the ventral furrow. So remember, we're talking about the ventral part of the embryo. The ventral furrow looks like this. If you look on the right side of the image, these are those mesoderm cells, those dorsal, high levels of dorsal twist, snail positive cells moving to the inside of the embryo. So you've taken this layer of cells, I told you it's superficial cleavage, so these layer of cells on the outside of the embryo with the yolk in the middle, and now you're beginning to form the mesoderm by bringing in some of those cells inside the embryo proper. And that's what this looks like. The ventral furrow is here, and this is a beautiful movie. You can see how complicated these cell movements are, and how actually it's very pretty, right? How pretty this process really is. So that's mesoderm. You formed mesoderm. Congratulations, you now have mesoderm. So I'm gonna stop this video. So this video has a really important point, and that's to tell you gastrulation is complicated. We've talked about one tiny part of Drosophila gastrulation. We're going to talk a lot more about gastrulation in this lecture and really in the next lecture as well. But I want to show you, I want to start by showing you gastrulation and how beautiful and complicated it is. And then at the end of these two lectures, you'll have a better understanding of gastrulation. And hopefully you can look at this video and say, I know exactly what's happening in this video. All right. So here's gastrulation in the fly. It starts with ventral furrow. Ventral is down here on the top image. It elongates. We'll talk about that process. These cells look like they're just sitting around doing nothing. They're doing crazy amounts of things right now. They're patterning themselves. You'll start to see these stripes form. Remember Eve stripe 2 when we talked about enhancer bashing? So these stripes are forming. And eventually that thing that elongated begins to retract back down, right? It's moving back down this way. And you see these segments are even more beautiful and more clear now. And then eventually this thing will do one more cool little thing and the video will be over. So we're going to hang out and let that cool thing happen. So you can see it's kind of elongating at the head. So we're forming a head. We're forming a tail down here. We are closing up. This is called dorsal closure. So the dorsal side of the embryo is zipping itself up. And then this thing elongates just a little bit more. And eventually this embryo hatches and pops out of this embryonic case and forms a larva. So the embryo turns into a larva in the fly. There's the heart. Heart's beating. Beep, 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 beep. It's beautiful. All right. I think this thing shows the larva hatching out. Doesn't show it. Oh well. Well, we waited for something cool and it didn't happen. Actually, lots of cool things happen. All right, so there's the beginning stages of gastrulation in the fly. It is beautiful, and it's really super complicated. So I just told you gastrulation begins with formation of the ventral furrow. These mesoderm cells are specified first and moved to the inside of the embryo. Here's another view of it. So these red cells are the presumptive mesoderm. You see them starting this invagination process. They form a tube on the inside. So you've got the mesoderm. Okay, mesoderm is formed. Next, the endoderm invaginates. So this is a type of cell movement called invagination. The endoderm invaginates in two places in the embryo, an anterior and posterior region, and these are anterior and posterior to the ventral furrow. So here's where the ventral furrow will be forming in the mesoderm. This is one region where the endoderm is invaginating, and this is another, this part right here. And you'll see that this has pole cells. Those pole cells have now turned into germ cells. And the germ cells are hitching a ride on the endoderm as it moves inside the embryo. And so here I'm going to show you two views. One, this is the ventral furrow, so we're looking at the ventral part of the embryo. Here it's called the anterior midgut invagination of the endoderm. Here I have the posterior, so we flip, this is ventral, this is dorsal view of the embryo. Here is the posterior midgut invagination of the endoderm, and along with it, it's the pulse cells. They're hitching a ride inside the embryo on this endoderm invagination. And then I also put a note here that the germ band retracts. So if you take a look at this, anterior, posterior in this top image, this yellow part, the endoderm, actually moves up, and you saw that in the video. It actually moves up to this part of the embryo. So you see the yellow moving gradually more anteriorly here, and eventually it's hanging out up here. So what really is the very tail end of the embryo is actually almost up next to what is the head of the embryo, because of this elongation of this structure called the germ band. And it's called the germ band because the germ cells are riding this wave. Eventually this thing retracts back down, and so the tail of the embryo ends up at the posterior part of the embryo instead of up next to the head. So this is what a germ band looks like. Here's the head. These are head segments. You now have a thorax segment, so segmentation is happening. And these are abdominal segments. So flies have 8 abdominal segments labeled 1, 2, 3, 4, 5, 6, 7, 8. So if I was to draw from head to tail on this embryo, here's the head, I'm moving, I'm moving, I'm moving, and here's the tail. So it's actually looped around on itself. You can think of it like a little chicken in an egg. It's like chicken within an egg, I mean. So it's kind of like wrapped up and tight on itself. So this is the butt of the embryo. This is the head. They're right next to each other, but really, linearly, this is how the structure looks. This germ band, this structure here, eventually retracts back down and comes here where it's supposed to be, the very posterior of the embryo. And segmentation is happening. So, importantly, the body part, or the body plan of the embryo, larva, and adult each have a distinct head, tail, middle region, and the middle regions are constructed of these repeating segments. We're going to talk a lot about how those segments are formed in the next lecture. So these adult segments can be traced right back to the embryonic segments. So without these embryonic segments, these adult segments don't form. So I'm going to show you this video one more time, and I want to pause it at important points. Alright, ventral furrow has formed. Here in this view you can see the germ band, so this is posterior, this is anterior, so the posterior part of the embryo actually will migrate up to here. Okay, so we're going to watch that migration. It's migrating up. Boop. If I can rewind just a bit. Right here are germ cells. And they're going to get, when this moves in, and you actually see that movement, when it moves inside the embryo, those germ cells move in on that wave. Okay? So this is the germ band. This is the very end of the embryo, but it's curled up upon itself. If we wait just a little bit, you'll see this germ band slowly start to retract back down. So this is the head. This is the posterior tail. And you start to see things kind of moving a little slowly, and then it kind of speeds up. So here's the tail. Here's the tip of the tail. I'm going to follow it with my mouse. The tip of the tail moves right back down here where it's supposed to be. So the tail's here now. And now you have this open yolk. The structure here is all yolk. And the embryo has to close around that yolk. So if you watch here, you'll see these cells begin to slowly migrate up. See them moving? These cells are migrating, and they're closing over the yolk and zipping up along this axis. This is called dorsal closure. So you're looking at the dorsal side of the embryo closing up. Okay. So I want to ask you a question. Pretend bacteria undergo gastrulation. True, false? The answer is false. Bacteria are not animals. They're prokaryotic. They don't have a nucleus. They don't undergo gastrulation. They're not even multicellular. It's a silly question. Okay, I'm not going to cover that today. I'm going to end here. Thank you very much for allowing me to give you a lecture on Blackboard. Again, I'm sorry I couldn't be there today. I really don't feel great, and I don't want to get you sick. We will talk next lecture about the exam and its results, and allow you to ask questions if you have them about the exam, so come prepared. And again, thank you very much, and I'll see you Thursday.